How is everybody? Everybody doing all right? 1 week, and then you get a long weekend. Last weekend before the big weekend. Um, all right. So before I dive in to protein denaturation, trying to make a wide arc, swing back towards food a little bit more concretely, I'm just going to talk about the final exam for next Friday. I have not written it yet. My inclination is to make it a multiple choice test, and I'll tell you why. That's my inclination. I want to experiment with these things called if-at forms. Has anybody used an if-at form yet in any of their classes? Well, they should be fun, I think. So it's going to be a multiple choice test, I think. You will take the test, you will hand it in on paper, I'll grade it, and then you'll break up into groups of a size that are as yet to be determined, and you'll take the test again. And you'll do it with these scratch-off forms that I have, like a lottery ticket, where you, as a group, talk about the question, and you scratch it off, and if you're wrong, then you can scratch off the next one, and you keep going till you get the right answer, all right? And then you progress to the next one, and so forth. So you'll get points for your initial attempt solo, and then that will be added to your group score that you'll have from the if-at form, all right? And the way that the if-at form works is, say there are four questions, four options on a particular question, and you scratch off one of them and you get it right, then you get points for all the remaining unscratched bubbles, right? So you get 3 points for that one, and if it takes you 2 bubbles to get the right answer, then you get 2 points, and if there's one unscratched one, then you get 1 point. And obviously, if you have to scratch them all to get the right answer, which I severely doubt, then you wouldn't get any points. But that would be added to your individual score. So the idea here is that it lifts your grade. It's probably going to lift everybody's grade, and you will know the answer by the end. So it's kind of summative and formative. I have a summative component, but also a formative. You've learned the material. As to what the material actually is, it's going to be just stuff from the lectures. I quiz you on the podcasts, so I'm probably not likely to ask you any specific question out of a podcast unless it directly dovetails with something I talked about in lecture. Okay? And when thinking about what material to think about when you're going through your notes or going back listening to my lectures again, I'm not here to bust you on super minute details. So I'm not looking for some obscure fact that was in the corner of a slide that I flashed through, or I often color my lectures with weird historical details. I'm not looking for that kind of thing. I'm looking for basic concepts, general concepts imagine being me making the test when you're studying for it. What kinds of things would you imagine are important that I'm trying to drive home in the basic study skill way of breaking the material down, okay? Does that make sense? Yeah, happy. Are we going to have lab on the day of midterm? I think so. I have one in the schedule, but I have yet to determine whether that's the case or not. I'll give you an answer to that this afternoon. I'll look at the schedule some more. I haven't decided. I have a lab set up, but I may throw it off. I imagine everybody would rather not have lab before fall break. Does anybody really want to have lab? It'll be fun, whatever I do. Okay, well, I'll think about that. Any other questions? Yeah, Hannah. I would say general ideas. If there are going to be people mentioned, I will probably mention them in the question, not asking you to regurgitate their name. You know, like Hennig Brandt discovered phosphorus how, or something like that. You know, like. Probably not. Probably not that. Yeah, that's like a. I mean, that's not a minute detail, but that's. Yeah, that's kind of putting you on. I mean, that's testing whether you know who Hennig Brandt is. I'm more concerned with what you know about the chemistry or the biochemistry or the science behind it. It's not. I don't want. I'm not giving you a history test. There's history that I can use to teach the science, but I'm not testing your history. Does that make sense? Okay. Good. Any other questions? Yeah. I haven't decided. I may decide after making the test and looking at it, or I may decide after I get the results back. But whatever, if that's the case, whatever decision I make will be to the benefit of people, not the detriment of people. So, yeah, I'm here to help you guys. I'm not trying to. I mean, I do want a grade distribution of some sort. I would be stuck if everybody had 100, but you know, I want you guys to learn the material. That's all tests are, is a way to like get you to learn the material. If every student was ideal, they would just learn material, and we never need to test you guys because you would be altruistically eating the material up, right? So, all right, that's enough pontification about testing. Protein denaturation. So last time I talked about what proteins were, how they got built out of amino acids, how peptide bonds formed, and the different structural elements that helped a protein fold into its native or active state. So now the process of digestion, eating food, is undoing all of that architecture that we so finely crafted. Okay, and you can think about this in terms of protein denaturation, and then preparing for protein digestion. We'll talk about actual digestion of protein or breaking the peptide bond when we get into the GI tract. This is not that. This is unfolding the protein to prepare it for digestion, okay? So denaturation. It's a disruption of these interactions, these side chain interactions, or these hydrophilic interactions that help keep a protein together, a disruption of them so that the protein is now inactive. It doesn't do what it was supposed to do. Proteins can be structural, they can be enzymatic. We'll talk about the enzymatic activity of proteins after break when I talk about coenzymes and micronutrients because those are related. So a denatured protein can either completely unfold or just kind of shift its structure so that it's no longer active. All right, there's a gradient of structure, functional but and folded, non-functional or inactive but still partially folded. Maybe secondary structural elements like beta sheets or alpha helices are there, and then fully denatured, a long chain. All right, so here are the principal methods of denaturing proteins and some examples of them from culinary sciences. So there's 5 main methods that humans denature their proteins. You can change the solvent that they're in. What's the typical solvent that proteins find themselves in? What's the principal solvent of life? Yeah. Cells are the containers of life, but...
Salt.
Huh?
Salt.
Oh, salt, pardon me. I can't hear over that fan. Salts are themselves solutes as well. The things that are dissolved in the solvent. What's the solvent? Yeah. Water, thank you. We live in an aqueous environment. Proteins live in an aqueous context. And so just change the solvent. There's a mezcal worm, a worm in the bottom of a bottle of mezcal, which is like an alcoholic beverage related to tequila. And the worm, those proteins in the worm get denatured by virtue of being soaked in alcohol. Has any of the 21-year-olds in the class had mezcal worm before? You can eat them. Looks of minor disgust. You guys are all so good. How about ceviche? Anyone had ceviche before? Yeah, a couple of you. What do you think of it? You like it? Never had it. Ceviche is not cooked. It's not heat denatured protein, but the protein has been denatured by changing the pH. You take raw fish or shrimp, or you can make all kinds of different ceviche, and you hit it with vinegar or lemon juice, driving the pH down and denaturing the protein. Another method is adding ionic substances, adding salt, the salts that Christian was talking about. I have salted meat over there, so cured meat, salted meat. What's another example of salting something to preserve it, to denature the proteins, change the proteins that you guys are very familiar with, that we're going to be dealing with? Sauerkraut. Okay, another example. All right, so obviously we denature proteins by raising the temperature. So there's a big chunk of grilled, sizzling meat, a T-bone steak on the grill there. Maybe a little more appetizing than a mezcal worm. Maybe not for some of you. And then the last is violent agitation. Just physically smashing the protein. Smashing it. An example are whipped egg whites. You can whip up some egg whites and make a meringue. Anyone ever made, or any like avid bakers in here, cooks? I've asked a few of you that before. Yeah, have you ever made a meringue? Go to it. Get it. What are some other examples? Can anyone think of another example of violent agitation? I'm not talking about protests here. Political protests, yeah. So what is that an example? Which type of denaturation is that? Cooking an egg. Frying it. Huh? Number 4. That's it. Raising the temperature. I'm looking for an example of 5. Anyone think of another example of that? Yeah. Thank you. You read my mind. Whipping heavy cream. Exactly. You just whipped cream. It's milk. You whip it. You denature the albumin proteins in there, and you form an emulsion is what it is. We'll talk about emulsions in lab in a few weeks. All right. So there are the methods. Let's see how these work in detail. Okay. Changing the solvents. So what happens by changing the solvent that you change from typically is going to be a little bit less hydrophilic than water. So ethanol is a little bit amphiphilic, meaning it has an OH group. Let me write the structure down. Anyone know what ethanol looks like? Water. Ethanol. All right. So it has this part over here that is hydrophilic like water, but then it has this chunk over here that is hydrophobic. Small hydrophobic tail. Not much of one. So what this is able to do is disrupt the hydrophobic interactions that normally sequester themselves to the interior of a protein, and now they can come to the surface of a protein a little bit. We can change the structure, slightly disrupt the structure of that protein. So now we see in the right-hand side, some of those hydrophobic amino acids depicted as little white balls on the chain there, they're on the outside of the protein because they're associated somehow with this tail of the ethanol molecule, but they're being shielded from the water by this hydroxyl group that's sticking out. All right, so ethanol has disrupted the native structure of the protein. You guys get that? Pretty straightforward. All right, next is changing the pH. So I had talked about various ionic interactions, charged interactions, interactions between basic and acidic amino acids, side chains, on different parts of the protein, maybe very disparate parts of the protein, but they're attracted to one another and they come together and add some structural integrity to the native state of the protein. Well, if you change the pH, you're going to change the protonation state of either of these acidic or basic amino acids. So, for example, if we lower the pH of the solution, that carboxylate group in aspartic acid gets protonated, and it becomes it goes from aspartate, the conjugate base, to aspartic acid, right? And so now it's no longer positively charged. The lysine stays protonated. It stays protonated because we're at a low pH. There's lots of protons in solution. We're not changing the charge on that, but it's no longer attracted to that negative charge on the carboxylate group. All right? So that gets torn apart. Also, for example, if we have two aspartic acids right next to each other, at neutral pH, they're both negatively charged. They may be pushed away from each other. That may be important to the native structure of the protein. However, if they get protonated, we can suddenly have a hydrogen bond interaction between them. That's going to also potentially disrupt the native state of the protein. All right. So at low pH, at low pH or in acidic environments, acidic amino acids are going to get protonated. All right, and that's going to throw off some of these charge interactions. Conversely, at high pH, we have the exact opposite thing. The basic amino acids are going to get deprotonated because at alkaline or basic conditions, there's a lot of proton scavengers, a lot of proton acceptors in solution. So they're going to tear any kind of free positive charge in the form of protons off of any exposed amino acids. So for example, lysine at a normal pH, which would be protonated at neutral pH or physiologic pH, would be protonated, and it would be attracted to the aspartate carboxylate group, the negative charge on the carboxylate of aspartic acid. However, if we increase the pH, make it more basic, more alkaline, these are all synonyms, the lysine will get deprotonated because some base, the hydroxide in solution, will pull off that proton, and the lysine and the aspartate will not be as attracted to one another as they once were. Okay, so again, throwing off the native state of the protein. You guys following me? Pretty easy. Okay. Here is the salted pork. So again, it's just a disruption of these charge-charge interactions. We have, again, this aspartate and lysine that are attracted to one another because they have opposite charges. Now we dump a bunch of sodium chloride all over it, and the sodium shields the carboxylate anion, and the chloride shields the cation on the lysine. So we have opposite charges attract to one another, and these free ions from the sodium and the chloride are shielding the positive and negative charges on the aspartic acid and the lysine from interacting with one another. Again, denaturing the native state of the protein and changing its activity. You guys with me? All right. So here's raising the temperature. Here's Christian's egg. No, was it Nathan's egg? Whose egg? Someone had an egg. Christian, you had an egg. Yeah. Well, here it is. You have the active proteins. These are the albumin, which is the egg white. The egg yellow, the yolk, is actually mostly fat, but the egg white is albumin. You heat it up, and that protein strand completely unfolds. And what happens is, as it unfolds, it's sticky, and it cross-links with other unfolded strands of albumin. And it cross-links and forms permanent bridges between each other, all right? And that can't be undone. And this has to do with the specific amino acids that are in the albumin and the ability of them to form cross-linked bridges in the albumin. All right, so that's a fried egg. So you can't unfry an egg once an egg has been unfried. However, some proteins act differently when they are heat stressed or heat denatured. For example, the protein that you'll find in milk, warm milk. So if you heat, has anybody ever had their grandma or their mom, you can't sleep at night, and they heat you up some milk, right? Really, really comforting. Or I'm sure you've all had hot chocolate before made with some milk. You heat the milk up and you bring it just to a boil, and then you let it cool down. You have absolutely denatured those proteins when it comes to that gentle boil. But as you let it cool down, and if you let the milk cool all the way down, you drink it, it doesn't taste any different. It's not like the milk has congealed into a solid mass the way an egg protein has. The protein refolds itself. It's able to refold itself in the milk. All right? Again, specific to the unique characteristics of those proteins. And now, you warm up milk for a baby so that it's at body temperature. So when you warm up milk, you don't take it to scalding or anything. You just warm it up in warm water secondarily, and it'll drop on your thing so that when they drink it, because babies evolutionarily would be breastfeeding, and they're getting the milk at 98.4 degrees centigrade, right, which is body temperature. And so they bring it up so that the child is to that temperature. That's why. Any other questions? We could talk about this a lot more, but yeah, Sonny, and then David. You'll have to be so strong. I'm sorry, I can't really hear. Yeah. There's a skin that forms on the top of milk. Not all of the protein in milk is the same. It's not all just one protein. Some of the proteins in milk do denature irreversibly, and that's the skin that you see forming on the top of that protein. Okay, and I mean, and this is what cheese is. Has anybody ever made cheese before, like paneer? Does anybody like Indian food? A few of you. You ever had paneer? The like Indian cheese, the Indian white cheese. Really easy to make. You just take milk, you heat it up, and then you squeeze some lemon juice in it. You squeeze lemon juice in it, and you immediately, it's amazing what happens. You get the curds and whey. Little Miss Muffet sat on a tuffet eating her curds and whey. It separates into these cheese curds, which are basically big blobs of denatured protein that crash out of solution, because once you heat the protein up and it becomes denatured and open, then you hit it with acid and change the pH. You're revealing amino acids in the milk when you heat denature it, and you change the pH. You're changing the proteination state of the acidic amino acids in the milk protein, and then you cause it to cross-link and agglutinate and form these curds. And what's left over is the whey. The proteins whey is in fact full of protein. There's lots of protein in whey. Those are the proteins that still do not agglutinate after denaturation. No one made curds and whey before? Yeah. You gotta say it louder. If you squeeze warm lemon juice out. No, you just need to squeeze enough lemon juice to change the pH, and once you change the pH enough, then you're going to get, I mean, if you just put a drop, you're not going to change the pH much. You may get some agglutination. So I guess there is a titratable curve there. Yeah, we should do that in class. It'd be fun. Make cheese, curds, and whey. You want to do that in lab? Okay, so yeah, here is irreversible denaturation. This is how this happens in Christian's breakfast egg. So we have the native albumin. There are all these SH bonds in the amino acid. We denature it, and then they form disulfide links that cross-link the protein. There's not a lot of sulfur in breast milk, but the sulfur that's in eggs is why eggs start to smell bad when they rot. It's hydrogen sulfide gas being given off by the bacteria that are starting to break these proteins down. All right, it's also why farts can smell a little gross and eggy sometimes, as you guys remember from the Brains On podcast about fart smarts. All right. So it's this process of raising the temperature to get irreversible denaturation, which is at the core of heat sterilization. So this is an autoclave. Who here has a piercing of some sort? Or a tattoo? Piercings or tattoos? Yeah, so particularly tattoos. I guess piercings, they take new needles out. And I guess they do in tattoos as well. They don't really autoclave in those. I mean, they do autoclave some stuff. This is how you, like if you go to have surgery, they will autoclave all of the medical equipment that they're going to use on you so that if it has ever come in contact with any bloodborne pathogens of any sort, that's all been autoclaved out, denatured, and destroyed by extremely high pressure, high temperature. What? High pressure? What are you talking about, high pressure, Dr. Klepak? Well, again, this takes, I should put a picture of the Woodsman Club sign up on this slide, because PV does, in fact, equal nRT. The pressure and volume equals the number of molecules in the gas times the temperature. So by varying pressure, you can affect temperature, right? And this chart here shows the relationship between pressure and temperature and the native state of a protein. So a protein can stay in its native state, and you can raise the temperature, but the exact temperature of denaturation is going to change based upon the pressure that you're at. Right? So down here at low pressure, this temperature will not denature. But at a higher pressure, it will be. For example, here, at this pressure, this temperature will not be. Whereas at this pressure, that temperature will be. You guys follow that? So pressure and temperature can also denature proteins. That's the concept behind a pressure cooker. Has anyone ever used a pressure cooker before? Pretty incredible to cook things fast. You have? Yeah? Cool. All right. So proteins get digested into individual amino acids, and they don't have any of their activity. After they get denatured, they get digested. We'll talk specifically about the chemistry of that in a few weeks. But once they get digested, or denatured by the stomach acid and then digested, they don't do what they're supposed to do any longer. They just become a source of amino acids. For example, this is why insulin, diabetics who need to take insulin, need to get it as an injection rather than a pill. If you were to take it as a pill, it would be denatured and destroyed and inactive. All right? So there's got to be some dietary sources of amino acids, and there's a big panoply of food. We have legumes of various sorts, beans, soy, cheese, fish, chicken, nuts, meat, eggs, milk, lots of different choices for people. These amino acids are what the body uses to synthesize upwards of 140,000 different proteins. Not different structures. I mean, in many trillions of copies, right? So your body's making trillions of proteins every minute, but 140,000 different proteins made up of these 20 amino acids. So, of them, the ones in red, circled in red, are essential, meaning that you have to get them in your diet. The body can synthesize all of the others. You can synthesize all of the others out of some other amino acid or out of intermediates from the metabolic pathways, central metabolism that we'll talk about. But animals cannot make the essential amino acids. Two of them are made insufficiently. So arginine and histidine are made insufficiently. So here are some recommended daily allowances of protein for body mass and gender, and also for women, whether they're pregnant or lactating. These values are amounts of protein per day per kilogram of body mass. However, the values are given per day, but honestly, if you don't meet a protein requirement in a certain day, if you get more the next day, it kind of averages out. I mean, it averages out across a broader stretch of time, all right? And you can see that as you get older, you need a little bit more protein just to be able to maintain the requirements for the amino acids. If you're pregnant and lactating, you also need a little bit more protein because you're building the fetus and you're putting protein, quite a bit of protein, into the milk. So when we look at a little bit more fine grain, a higher resolution of this data for the specific essential amino acids, you can see that there are some significant requirements here. Again, this is listed per day, but if you don't get 100 and let's look. For adults, you would need 12 milligrams per kilogram of body mass per day. If you don't get that much lysine in a day, you're going to be all right because you'll have some lysine in your amino acid pool, your nutrient pool in your body, and you'll make it through. Without too much problem. So here's an important concept: nitrogen balance. And I want you to understand this concept because it'll help you assess information that was in the podcast for this week. So nitrogen balance is a measure of how much nitrogen you're consuming in the form of protein ingested versus how much nitrogen is leaving the body as you excrete it through the urine and some of it in your feces. All right? So a person like me who is not putting on body mass and who's not sick and is not who's just living his life, I'm in a neutral nitrogen balance. The amount of protein I eat is the same amount of protein that I excrete. All right, what comes in is essentially going out. Now I'm absorbing some and assimilating it, and then my body is sloughing some off and removing it in the urine or in the feces. All right. However, people that are in what are called positive nitrogen balance are people whose absorption through ingestion is more than they are excreting. So examples can be bodybuilders, people that are putting on body mass. If you're not putting on body mass, you're not in positive nitrogen balance. Pregnant women who are putting on body mass, growing protein. Children, people who are growing, that includes you guys. Many of you are still at the tail end of adding muscle mass to your body from your growth. And so you guys are all probably in relative positive nitrogen balance right now. Negative nitrogen balance. This is where your excretion exceeds your ability to absorb protein. And there's a number of examples of this. On the left is the sad case of anorexia or bulimia. These are people who are just not eating enough protein, and their body, they're not getting enough calories. They're burning protein for energy, and the nitrogen is leaving in their urine through urea. People who are extremely malnourished because of famine. So that's what you see over there. Kwashiorkor, this poor child there. And then people with wasting diseases. So there's Stephen Hawking, who has Lou Gehrig's disease or some kind of thing like that. And at least for a while he was losing body mass, he was in a negative nitrogen balance. All right. So there are complete proteins. These are proteins that contain all of the amino acids in the correct proportion for humans. So all animal products are going to be complete proteins. And there are several complete proteins in the vegetable kingdom. So quinoa, hemp seed, amaranth, soybeans, buckwheat, chia, and peas. These are all vegetables that have all of the essential amino acids in the right proportions. I want to stress that because it's not just that. So virtually all, I would struggle to think of one that doesn't, but virtually all vegetable protein sources have all the amino acids. Just some of them are very, very trace, and not a reliable source of those essential amino acids. These vegetable sources are complete proteins in that You could live just off of quinoa if you wanted in terms of your protein requirements. Nobody ever would, okay? But and then introducing the concept of incomplete proteins. So these are foods that have one or more of the essential amino acids in a very low quantity, a very low proportion of the overall diet. So, for example, wheat and rice lacks lysine, peas and beans lack methionine. So this diet of rice and beans combined is an excellent long-term source of complete protein. All right, complementary. It's the notion of complementary proteins. Another example here is hummus, which has garbanzo beans and sesame seeds, both excellent sorts of protein, except lacking some methionine. And then pita, which lacks lysine. All right, so you put those together, and together you have complementary proteins that are giving you a complete nutritional profile. So what's interesting about these two foods, these meals were established centuries before people ever understood the notion of complete or incomplete proteins, and probably evolved over time as people just experimented and found foods that were healthy, a healthy source of nutrition. They evolved in cultures that had probably a much more homogeneous diet than any of you have. Any of you have. Everybody has probably in this room has a very diverse, varied diet that has multiple protein sources from vegetables. All right. So, who thought up food combining? Was this woman, Frances Moore Lappé. I'm going to tell you the story about her on Monday. I'm going to pick up with her on Monday because I want to go and take the quiz with you guys. And so I only have a couple more slides, and we can talk about this woman and her lovely family next time. But whip out some paper. Yeah, I'll tell you the story of her next time.